In this module, I will discuss about post-transcriptional modification of tRNA and genetic code. Now, post-transcriptional modification of tRNA is described in this illustration. RNA polymerase III, it transcribed tRNA. Now here in this figure, the figure in the brackets indicates nucleotide base pair. The transcript which is produced is about 108 base pair long. Now 5' trimming first occur in 3 steps at 104 base pair, 97 base pair and 92 base pairs. There is a 14 nucleotides intervening sequence. So 5' trimming starts in the leader sequence and 3' trimming starts at the trailer sequence, and ultimately a CCA molecule is added at the 3' end. Finally, splicing occurs and mature RNA is released with the CCA end in the cytoplasm. Now let's see what are the important enzymes involved in post-transcriptional modification of yeast tRNA or eukaryotic tRNA. Now tRNA normally has intron which also varies from 14 to 60 nucleotides. Now 5' trimming occurs by an enzyme, it is called RNase P. Now this RNase P, this enzyme consists of M1 RNA and a polypeptide. Now 3' processing carried out by 6 RNases and synthesis of CCA at 3' terminus carried out by an enzyme tRNA nucleotidal transferase. So tRNA and also one ATP molecule is required to carry out these reactions. So tRNA plus ATP plus 2 molecules of cytidine triphosphate. In the presence of tRNA nucleotidal transferase, a CCA end is added with the tRNA molecule. Splicing of intron is carried out by RNase A and Q1 enzymes. Genetic code. So what is genetic code? The genetic code or codon is a nucleotide triplet. So nucleotide triplet is called genetic code. There are 64 codons which code for 24 amino. There are code for 20 amino acids. So there are 64 codons which code for 20 amino acids. Now genetic code is degenerate, that is multiple codons are there for a particular genetic, for a particular amino acid. So it is called genetic code degenerate. Code is comma free and non-overlapping. Moreover, it is start and stop codons are there. Genetic code is universal, that is genetic code is same for almost all the individuals, that is for prokaryotes, eukaryotes and all animals, individuals of the animal kingdom. Now AUG, it forms the start codon, that start codon is AUG, while stop codons, there are 3 stop codons are there, UAA, UAG and UGA. UGA. So there are 3 stop codon and 1 start codon is there. So how the codon anticodon pairing occurs? Now FSC Crick, he has given one hypothesis regarding codon anticodon pairing. According to that hypothesis, that is, the first 2 bases of a codon must pair correctly according to Watson-Crick pairing rule, but third base of the codon can wobble from its normal position to form unusual base pair called wobble base pair. So this wobble base pair, it solves the problem because it reduces the number of tRNAs required to translate the genetic code, because there are 64 codons are there and there are less number of tRNA. So wobble pairing reduces the number of tRNA required to translate a genetic code, and it solves the particular problem of genetic decoding. Now, as for wobble pairing, there is some pairing rules are there. That is, G will pair with U or C, C pair with G, A pair with U, and U pair with A or G, and ionosine molecule is there. It is paired with I. Ionosine or I, it pairs with A, U or C. So this wobble pairing occurs, that is the first base of anticodon in 5' position of tRNA, it pairs with third base of codon in the 3' mRNA. So this is called wobble pairing, and this pairing, as per these rules, it occurs. So in these diagrams, it has been shown that is the how the in the 1 position, that is of the anticodon, and 3 position of the codon. So 1 and 3 here wobbling or wobble pairing occurs. Now the structure of the tRNA, it is very interesting. Robert Hole, he first described the Cloverleaf structure of tRNA and how codon anticodon pairing occurs. This Cloverleaf structure of tRNA, it consists of a CCA acceptor stem and a 5' end. Then there is a T loop. It is also called T pseudouridine CG loop. Then there is a D loop. It has got a dihydrouridine base is there. Then there is also variable loop, and there are some rare bases are there which are included during post-transcriptional modifications which are inserted into this tRNA. So these are some of the rare bases, that is dihydrouridine, T pseudouridine, CG, etc. Now there is one anticodon loop is there. In the anticodon loop, so codon will come and there is a pairing occurs, that is the third position of the codon, which is called the wobble position. This called is the wobble positions. It binds with the first position of the anticodon, and rest 1 and 2 per position of the codon, they normally follow the WC pairing or Watson-Crick pairing. So as for example, we can show, suppose if AUG is the starting codon, then the anticodon will be UAC or UAU. So this is the way by which codon anticodon pairing occurs and decoding of genetic information takes place. So there are 5 scientists who has made a enormous contribution towards the decoding of genetic informations. They are Robert Hole, F.H. Crick, Hargobind Khorana, Nirenberg, and Okwa. So all of these scientists plays an important role in solving the problem of decoding of genetic informations, and all of them received the Nobel Prize for this great milestone in the science of genetics. In this module, we have provided key concepts and summary. Books are also provided, list of books are provided for further reading. There are some quizzes are there and also study material. Thank you for attending our online course. Over and above, if you have any question, please feel free to ask us.